We'll continue our conversation about biotechnology today. This time we'll take it a step further and we'll go into agricultural biotechnology. But before we do that, I wanted to wrap up some concepts related to transgenics and knockouts in research. So this slide summarizes everything that we discussed last week, how you go from a specific target gene that you would like to replace in a mouse, how you recombine a transgene of interest into that target sequence in the mouse genome. You do that by transfection of embryonic stem cells. You select for those cells that will permanently incorporate that transgene of interest into its genome. Not all of them will, but the ones that do incorporate the transgene of interest will also incorporate some sort of selection marker, like resistance to a given antibiotic. You can then select for cells that picked up that transgene by killing everything else, and the survivors will keep proliferating in culture until you have many of them. Those transgenic embryonic stem cells proliferate, and then you inject them into an early mouse embryo. Those transgenic embryonic stem cells will hopefully intermingle with other embryonic stem cells in the inner cell mass, and it will contribute to that mouse that it develops from that embryo. And because there is a mixture of embryonic stem cells from two different origins, you get these things that we call chimeric mice. The trick that researchers use is to use embryonic stem cells from two different genetic backgrounds, one that carries alleles for black pigmentation and a background that carries an albino allele or a recessive for no pigmentation in the skin. And then when you cross those mice, we went through the explanation of why the mice in the project that you're interested in are those that are black, because those are the ones that potentially carry one copy of your transgene of interest. And if you have any questions about that, I refer you to the slides that we discussed last week, because all of the details are there. The point is that among this black progeny, from the cross between a chimeric mouse and a white mouse, there will be some that are homozygote wild type, so they don't carry any copies of the transgene of interest, but then there will be some, approximately half among those black mice in the progeny, that are heterozygote for your transgene of interest. And the easiest way to distinguish between these is by doing genotyping based on PCR. So you choose primers that flank both sides of your locus, insertion locus in the mouse genome. And if there was no insertion of a transgene, the PCR amplification product should be smaller. But if there was insertion of your transgene of interest and some sort of selection marker, typically that leads to a much larger amplicon. And obviously you can detect these two different types of amplicons based on size using gel electrophoresis. So if you have a mouse that, like the case on the left, is homozygote wild type, you should see only one band, because there's only one type of amplification product. But from one of these transgenic heterozygotes, you should get two bands, one small for the wild type allele and one larger for the transgenic. We took it a step further and we said that if you were to take those transgenic mice that you detected by PCR genotyping and you cross them between them, you are essentially carrying out a monohybrid cross, like a classical Mendelian cross between two heterozygotes. In this case, it would be heterozygotes for the transgene, and you might get lucky and you might get some homozygotes for that transgene. And you would detect them as those in which the only amplification product that you can detect is the long amplicons. Right? And that is because now both loci in this particular location have been replaced by the transgene and a selection marker. I posted a video in Canvas that I hope you had a chance to watch. If not, I invite you to do it. There is some useful information there. And if you did, you may recognize a version of this table. The video goes through how, depending on what is that we introduce into the mouse genome, the type of transgenic animal that we get out of this process, right? So what we discussed here in class are the classic transgenics. This is a very generic term to refer to animals in which we've put a foreign gene into their genome, right? And often the question that we ask for those classic transgenics is what happens if you express this gene in a mouse, right? So the approach in that case is relatively straightforward. You clone your transgene of interest just downstream of a regular promoter, sometimes a strong promoter like a viral promoter, but sometimes just a regular promoter, even the endogenous promoter for the gene from its source, and then you simply place it somewhere in the genome and hope that the mouse will start expressing it, right? But depending on what we put in these transgenes, all these kinds of different kinds of transgenics that we can get. Knockouts, we discussed them briefly last week. Mice that overexpress a given gene if you use a very strong promoter, tissue-specific expression of a gene, so you're not expressing it everywhere in the mouse, but in certain tissues. Inducible promoters that allows you to control the timing of expression. Reporters, these are very special transgenics that allow you to see where an endogenous gene, a normal gene, would normally be expressed. And the trick here is to use the regular promoter of a gene to drive the expression of something that you can see, like one of these fluorescent proteins or an enzyme that can turn a colorless reagent into a colored product, or anything that will allow you to visually detect expression of these things. The video talks about tagged genes and then knockdowns as well, where we make an animal in which we're not completely knocking out the gene from the genome, but we're simply introducing one of these RNAis or RNA hairpins that will make the microRNA machinery kick in and inhibit the expression of whatever you're trying to inhibit. I flew through this, but once again, there is a video posted in Canvas about all of that. Super briefly, we discussed how you make knockouts using transgenesis. The idea here is that instead of introducing a gene into a given location of the mouse, what you do is you target a gene that you would like to take out from that genome, and you replace it with nothing. Now, when I say nothing, I don't really mean it. What you normally do is you replace that gene that you'd like to take out with just selection marker. Say the resistance against neomycin gene, right? So you're targeting a gene that exists in the animal for something that doesn't exist in the animal. In that process of swapping sequences, you're effectively taking out a given sequence from that mouse, right? So in knockouts, there is no transgene of interest and no promoter, and all you do is you replace a given gene of the mouse with a neomycin-resistant marker. You go through the whole process that we discussed in the previous slide, and I think I have it here. So at the end of that process, again, you can end up with some mice that are heterozygote for the knocked out locus, in which the wild-type gene has been replaced by a neomycin-resistant cassette. In those heterozygotes, there will still be a regular copy of the gene that you wanted to knock out. But if you're lucky, you can cross these heterozygous mice to each other, and you might get some homozygote knockouts in which both copies of this gene have been swapped by the selection marker. A subtlety here that is kind of meant to reflect reality. Normally, when we're trying to replace wild-type sequences in the mouse genome for these neomycin-resistant cassettes, typically the endogenous gene that you're trying to replace is much larger than the sequence that you use to replace it with, right? So these neomycin-resistant resistance genes tend to be really small compared to the endogenous genes in the mouse. And why that is, it has to do with the fact that the endogenous gene will often have introns in it, so it's typically longer than the very small sequence that you need to just express this enzyme that will make the cells resist neomycin. So the things flip in the sense that now your homozygote knockout tends to give rise to smaller amplicons in that PCR genotyping, not larger ones, because you're not introducing something large in a given mouse genome location. You're actually replacing something that used to be large for something that is much smaller, okay? And that's shown here how those PCR primers that we normally design flanking the locus, the targeted locus, will now will probably give rise to something that is much smaller. So if we go through the same exercise and we cross two heterozygotes for these knocked out locus, and we look among the progeny, like if this cross will follow Mendelian ratios, you expect about a fourth of them to now be homozygote for the knocked out gene, right? So out of these 8 mice, it turns out that there is actually 3, well, you expected 2, but you got 3 that are homozygote knockout because they only produce that small PCR amplification product that you expect from the knocked out gene. So in this particular example, mice 2, 5, and 7 would be knockout for your gene of interest. And you can start asking questions about what is happening to this mouse that doesn't have that gene. Remember, this is all in the context of what we discussed or we introduced last week as reverse genetics, in which you start from a gene that you're interested in finding out more about, and then you start asking questions about what are the phenotypes associated with, in this case, its disappearance from the animal, right? So whenever you have a gene that you're interested in and you'd like to find out what it does in the animal, one of the most straightforward approaches is just take it out. Mutate it, delete it, take it out, and see what happens to the animal, right? Sometimes the effects are pretty drastic. You can see immediately that these mice are not normal anatomically, physiologically, behaviorally, but sometimes it's not that obvious and you need to subject these mice to a series of tests, and sometimes you need to look internally to see what might be wrong with them. And yet there are times in which we do the exact same thing and then you cross the heterozygotes, which you obtained successfully, and then you're hoping to get those homozygote knockouts in the progeny from that monohybrid cross, but you get none. There's no mouse in the progeny that has nothing but the small amplicon. So what do you think happened? A priori assumption we can make is that that gene that we took out was absolutely necessary for those mouse embryos to develop through to adulthood. So we never saw those pups, we never saw those mice in the progeny because that gene that we took out, they needed it, right? We call that embryonic lethality, and we had kind of seen this concept before when we were talking about Mendelian genetics a few weeks ago, and whenever you expected one of those 1 to 2 to 1 ratios, and then it turns out that you get a 1 to 2 only because there is one genotype that is missing, and it typically is the homozygote recessive. And the simplest explanation for that is that the individual needed at least one copy of that gene. And it can be a heterozygote, and it can be obviously a homozygote dominant or homozygote wild type in this case, but it cannot live without that gene, right? So often you can have adult mice that are knockout for a gene of interest, but very often when you try and do this, you don't get any viable adults because those genes were needed throughout development. So for a while, it was impossible to study these genes in a knockout context, right? You could not knock it out from a mouse because you couldn't get a living mouse out of this. So that led to this new concept, is more recent, if you will, of the conditional or inducible knockouts. And what that means is you will have a gene that is knocked out from cells in the animal, but only in certain cells of the animal or at certain times during the life of that animal. So there are conditional knockouts in the sense that certain conditions need to be met before those genes can be knocked out. And I'm going to walk you in more or less detail about how this works. You may have heard of Barbara McClintock. She won the Nobel Prize Physiology and Medicine for the discovery of transposons. Transposons sometimes go by the common name of jumping DNA. And these are DNA sequences that, like the name indicates, have the ability to cut themselves out from where they are and reinsert themselves somewhere else into a genome, right? We talked about these guys briefly when we talked about mutations and how there is a kind of mutation caused by the mobilization of these transposons, and how when these jumping DNAs jump around the genome, sometimes they will insert themselves and break important genetic information in the process, or sometimes in their process of jumping out of the genome, they will break some sequence around them and again cause some mutations. Right? So we did introduce transposons before, but very briefly, and once again, there is a piece of DNA that is somewhere in the genome, and that piece of DNA, that transposon DNA, codes, among other things, for these very peculiar enzymes called recombinases. And these recombinases can have the ability to detect certain sequences on both ends of the transposon DNA and then recombine them, induce one of these genetic recombination events, almost like a crossing over that doesn't happen during meiosis, right? So there is this swapping of genetic information, but instead of being between homologous chromosomes, is within the same chromosome molecule. So there is this looping of the DNA shown here, and then there is a recombination event that takes place, I meant to write there, a recombination event that takes place such that there is a loop of circular DNA that jumps out of the genome, and then these two ends are rejoined. So the site where the transposon used to be is reconstituted without a transposon in it anymore. And now this transposon will move somewhere else, and using the same recombinases coded by the transposon DNA, it will undo that process and insert itself in somewhere in the genome of the same cell. So this transposon didn't go anywhere far. It stayed within the same cell. It's just that moved locations within that DNA. Okay? Transposons are natural entities. These things have been evolving with multicellular organisms for way longer we've been around. Right? Transposons exist way before genetic engineering. But as usual, genetic engineers hacked a natural system, like transposons, to our advantage. And the way that this goes is as follows. It turns out that in time we learned more and more and more about this process. And we learned, for instance, that the enzyme that does this recombination, these recombinases that are expressed by the transposons, recognize consensus sequences. They won't induce a recombination of just any two sequences. As usual, they will recognize sequences that are actually on the longer side. They're somewhere between 15 and 20 nucleotides in length. So they're not going to go and bind just anywhere in DNA. They will look for very specific, rather long sequences that are in DNA and that happen to be those sequences that flank the transposon DNA, right? So there is different transposons. The one that we use for conditional knockouts uses an enzyme that is called CRE. And the consensus sequences that flank transposons and that we hacked for conditional knockouts are called LOXP sites. The history of that, I personally don't know it. I'm sure that there is a history to those names, but LOXP sites are the ones that are flanking the transposon and recognized by CRE. So CRE recombinase in the CRE LOX system will do this recombination business. And here's a slide that shows it in a bit more detail. So what happens is these two LOXP sites are near identical to one another, right? And what happens is when there is that looping of DNA that I showed before, CRE recombinase, recognizing those two sequences that are placed on top of one another, can now do the exchange of DNA strands, very much like what you would see when two sister chromatids synapse with each other during crossing over during meiosis, right? So there is this understanding by CRE that these two sequences are almost identical to one another, and that triggers the actual enzymatic event of cutting, inducing those cuts in both strands of DNA in an orderly fashion such that things are swapped, and as indicated here, you have all DNA followed by or sort of the DNA and the first half or so of the LOXP site, followed by the second half of the LOXP site from the second LOXP sequence in that genomic location. As I said before, this thing, the transposon, the circularized transposon, evolved to quickly jump and insert itself somewhere else. So if this transposon is lucky, it will evade that process of that defense mechanism that we discussed before, that eukaryotic cells will mount anytime they see some circular DNA going around. But because this loop is attached to these Cre recombinases, the whole insertion process somewhere else can happen quickly, and that transposon survived. So that's the natural life of a transposon. How did we hack it in the lab? So it goes something like this. Let's say that your target gene is here. It's indicated by that beige line. And we know that's the gene that we would like to take out from the mouse, but in a conditional manner. We know that this is one of those genes that if we take out using regular knockouts, we'll get embryonic lethality. So now we know better. We know that we cannot take it out through a regular knockout. We need to do one of these conditional knockouts. So what we do is, in the lab, we engineer a version of that target gene with its normal sequence. Everything is normal about this gene. And we purposely flank it with these LOXP sites. And then we flank both the what we call the floxed gene with a sequence that is on both those two ends, here and here, with sequence that is homologous to flanking regions in the target locus, right? Why do we do that? Because we hope that when we inject this whole engineered construct into embryonic stem cells, some of those embryonic stem cells will carry out that homologous recombination between the homology regions and swap our target gene for the whole thing. And when I say the whole thing, what do I mean? I mean, the target gene, we're replacing the normal gene with a new copy of the normal gene, except that this one that we're putting back in is floxed. It's flanked by LOXP sites. That's hence the name floxed, flanked by LOXP sites, right? And usually what we do, it's a little trick, we will also introduce a neomycin resistance cassette as usual, so that we can select these embryonic stem cells that incorporated this thing into their genome. And we also introduce a sequence for GFP. And if you guys remember, GFP stands for green fluorescent protein. And you'll see why we do that in a second. So we go through the whole process of making a transgenic mouse. This time we're not overexpressing anything. We're not creating a reporter. We're not creating a knockdown mouse. We're creating a floxed mouse. We're trying to introduce this transgene that has the same target gene that we wanted to knock out, eventually floxed or flanked by these LOXP sequences. So now we have our floxed mouse genome. Using the tricks that I showed you before with PCR genotyping, we identify those mice that are heterozygotes for this transgene, and then eventually, if we're lucky, we can get a homozygote floxed mouse. Then separately, we need to get our hands on a mouse that will express Cre recombinase under some sort of either cell type specific or inducible promoter. Now, these mice normally we don't make. These mice have already been made and companies will sell them to you, right? So you can go into a catalog and say, I would like a Cre mouse for expression in, and then you fill in the blank with your favorite cell type. It could be a neuron, it could be a muscle cell, it could be an intestinal cell, it could be whatever. Other times you say, I would like a Cre mouse that will express Cre when, and then you decide when that will be, when you feed it a certain drug, when the mouse reached adulthood, when the mouse is under metabolic stress, when the mouse is in a very hot environment. You choose a promoter that will respond to some sort of environmental condition such that you can control the timing of expression of that Cree. So these mice, all they will do is express the recombinase in either a cell type or a cell condition dependent manner. Okay? But they don't do anything else by themselves. The magic comes when you cross these two mice together. The Cree mouse that you purchased and the floxed mouse that you made. In progeny, you will have that Cree will be expressed either in certain cell types or under certain conditions. And when Cree is expressed, that Cree will go, and as shown in this part of the diagram, will go and induce that recombination of the LOXP sites, effectively taking everything that was between those LOXP sequences. And what was between those LOXP sequences? Your gene, the gene that you wanted to knock out. So once again, we do this because there is no, the gene is there until you take it out. So these mice can get through embryonic development just fine, and then Cree will be expressed in either some adult cell type or when you tell that mouse to express Cree. What's your favorite cell type in the organism? Brain, a brain cell, a neuron, right? So you could make these Cree, so you could choose a Cree mouse that will express Cree only in adult motor neurons in the hippocampus region C1. I'm making this up, but it's not far from what happens. You can go into these catalogs and find that mouse. What's going to happen then is that mouse will not express any Cree until it developed a brain with a hippocampus with some motor neurons in the C1 region. Only then it will express Cree, and only then Cree will go to work and take out your gene of interest from those cells only. And that's why the mouse can make it just fine through embryonic development, and they will make it at the very least to pups, right? Sometimes if these Cree will start getting expressed soon after birth, you'll have to hurry up to see what happens when you take the gene out of those cells. But at least you have a mouse where to look. And needless to say, sometimes the gene may be needed somewhere else, but when you take it out from these very specific cells that you're interested in the brain, you don't kill the mouse. You may be looking at very fancy, complex effects of taking that gene out, but you're not killing it. So that's why we call them conditional knockouts. There are certain conditions to be met before the gene of interest gets knocked out from the genome. So the way this works in real life is, remember how I mentioned that we introduced I made it sound like it was just for fun, but it's not. It's useful. We introduced a GFP sequence there. Guess what happens to that GFP sequence when we take out the FLOX gene? Now that GFP sequence is much closer to what used to be the promoter for that gene, right? So the GFP sequence was not expressed before you took out the FLOX gene, but now when you took out the FLOX gene, like here, now you have the promoter in close proximity to the GFP sequence, and you will start expressing GFP sequence. Why do we do that? Because then when we take this mouse and dissect the brain out, for instance, we will look at the neurons, and some of them will not express any GFP because this Cree recombination business is not foolproof. It's not 100% efficient, but some of them will. And those that express GFP, are cells that have lost your gene of interest. So now you can look inside of those cells and start making some conclusions about how they're operating, what they express, what they don't, what they look like, if they're big, are they small, do they have all the right branching, blah, blah, blah. All right, so now we can turn the page and start talking about agrobiotech. And I wanted to introduce this conversation with everybody, and I'd like to hear your thoughts. So bacterial cultures are great for mass production of biomolecules. There are many biotechnological companies that make a living out of selling small peptides that they produce in bacteria. You may have heard of recombinant insulin, recombinant growth hormone, recombinant this, recombinant that. Those are small peptides that are produced in bacteria in very large quantities, and they're great for that. But sometimes you do need eukaryotic cells. Why is that? Why would you need to express certain genes in eukaryotic cells? Why can't you express them in bacteria? The issue here is that there are certain proteins that in eukaryotic cells will go through a series of post-processing, if you will, of post-translational modifications that bacteria can't do. Certain decorations with certain sugars, certain modifications of amino acids with fatty acids, those things bacteria can't do. And there is a reason why eukaryotic cells carry them out. Often they're needed for the final function of those proteins. So there are many instances in which you simply can't express a eukaryotic protein in a bacterial cell and get the full functional protein, right? So fine, we'll make them in, let's say, mice or rats, right? So can we have them simply express the peptide or protein of interest, let's say in blood or milk or saliva, right? And they're eukaryotic organisms, they could be producing those proteins of interest for us. What might be a downside of taking this approach? They're very small. They're very small. So many of you may not have seen this movie. For those of you who recognize it, you recognize it. For those of you who don't, he's Ben Stiller describing how he would milk a cat in his farm when he was growing up. And the whole concept stands. They're too small. So if you are producing a given bacteria, a given human protein of interest in the mouse blood, for instance, you would have to euthanize hundreds, if not thousands, of mice before you can get enough. With bacteria, we can grow them in gigantic liquid culture tanks. That's what biotechnological companies do. Sometimes you can make these tanks for eukaryotic cells in culture, but if you were to try and do it in a living organism, a mouse, a rat wouldn't do it. So biotechnological companies for a long time have been interested in creating farm animals, cows, goats, sheep, chickens, that produce human proteins in their milk or eggs for two reasons. They're larger, and second, think the infrastructure of farming, right? It's huge. It's there already. So the supply chain, the caretakers for these animals, like you have everything you need to be able to produce these things at very large scale, right? So obviously the question becomes, can we make a transgenic cow, right? And the answer is yes. The only problem is that if we take the same approach that we discussed before for mice, if you guys remember, for mice, we went through 2 or 3 rounds of checking, right? So there was a first litter of mice that you would have to find the chimeric mice from. Then you would cross those to white mice and get those black mice that could be heterozygote for your transgene of interest. Then you would cross those between them and then look for those that are homozygote for your transgene of interest. In mice, that's relatively easy because they have very large broods and gestation is a little over 20 days. So within 3 months, you have your transgenic. Cows, for instance, will take almost a year to produce 1 calf, right? So not very convenient. Imagine the number of cows that you would have to first make the chimeric cow. A year later, you would cross it to a non-chimeric cow. A year later, it's just ridiculous. So with mice, you can play the numbers game. It's relatively easy. With cows, you can't. So what happened was someone came up with this idea of something called SCNT, which stands for somatic cell nuclear transfer. The idea here is that if you take an egg cell from a female cow, and you take the nucleus out of that egg, right? So you anucleate that egg, you end up with a nucleusless egg here, enucleated egg. If you now introduced the nucleus of any somatic cell that you get from a different animal, now that egg would be like a fertilized egg, right? it would have a nucleus that has two complements of genetic information, so it's diploid, and it has all the information that would have otherwise been in a regular cow. The trick then is you do all of your genetic engineering in a dish, because you don't need a year for that. You can do it in weeks. You get your transgenic embryonic stem cells. Instead of using mouse embryonic stem cells, you might use a cow embryonic stem cell line, or between us, any other cell line from a cow that you can relatively easily manipulate genetically. And then what you do, the trick is you take the nucleus from that transgenic cell that you obtain in culture and you carefully inject it into an enucleated egg. Now this egg thinks it's a zygote that came from fertilization. It never saw sperm in its life, but it still thinks that came from fertilization, and it will start dividing and make a cow or a bull. So yes, there is some time that you need to make those cells in the lab, but then within a year you will have your calf, and in a few years now you have an adult cow or bull that is transgenic for your gene of interest. Heterozygote, fine, because we're still in heterozygote animal, but at least you have it, and now you can have this animal reproduce many times, right? Now, if we have this animal reproduce with an animal that is not transgenic, what will happen? Well, half its progeny would be normal, because this cow is heterozygote, and half its progeny will inherit one copy of the transgene. So again, we get ourselves into this business of with farm animals. It takes a little longer to play this game of, I'll just cross them once and then pick up the heterozygotes and cross them again. It takes a little while, right? So what's the trick? The trick came from facilitating a little bit this whole injecting the nucleus thing. Imagine that egg cell that we get from the female, from the cow, when you take the nucleus out, most of the eggs won't make it, right? That hurts the egg. Some of them will. Some of them will survive that procedure. And now you're injecting them with a new nucleus to introduce a new nucleus into them, even worse. So it's a pretty violent procedure. In fact, somatic cell nuclear transfer exists since the late 60s, 1960s that is. So it's been around for a while, but it used to be done only in frogs. And the reason people could do it only in frogs is because the frog eggs are gigantic. So the materials that we use for doing all of this enucleation and reintroducing a nucleus didn't harm so much those huge eggs. But whenever you tried to do it with mammalian eggs, it was really damaging. So it was a while until Ian Wilmot in Scotland came up with a process for, instead of injecting the somatic cell nucleus into an enucleated egg, he would enucleate the egg, so that was still needed. But then he created a protocol for fusing the somatic cell with the nucleus in it still, and the enucleated egg. So now there's two cells fused, and basically now this fused cell, this fused hybrid cell, would have a nucleus. And a whole bunch of the cytoplasm from the donor cell would get transferred to this hybrid zygote, but this hybrid zygote still had enough information in it to know that it is a zygote and that it should grow into an embryo and eventually make an adult animal. And Ian Wilmot, for reasons that I personally don't know decided to do all of this with sheep instead of cows. But he established the proof of principle, and now this is why you hear these stories about people cloning their dogs or cloning farm animals or cloning their pets, right? It's real. Animal cloning is real. So when this technology became available and widespread, at first, like those things that make the news, people were like a little hesitant, but then everybody tried over and over, and it's real. It's real. So now, it was a lot simpler. You just take all the eggs you can from a cow, you enucleate them all. Yeah, some won't make it, but some will. And then instead of injecting them with eggs, killing most of them, you simply do this dolly-like fusion of a somatic cell with the egg, and you can create a hybrid zygote that can grow into a cow. The only thing you have to be a little careful with is what type of cell you use to be that donor of the nucleus, because that cell will have imprinted its genes. It will have laid down a series of epigenetic marks that will make the genetic information here often not be fully accessible to the zygote. So hence the stories that you may hear about when you clone an animal, the new animal that is born through this process is already old, right? Because there is a certain genetic age that gets imprinted epigenetically on this DNA. So by the time that the new calf is born, they're genetically a little older than a normal calf would have been. So the story goes, for instance, that Dolly, the sheep, died a lot younger than a normal sheep would, right? And now we know why, and people are doing a lot of research into how we can erase some of those epigenetic marks from cells that you can obtain way more easily from skin, from saliva, from muscle, right? Such that you can sort of run the clock back in those cells, epigenetically speaking, and when you clone the animal, it's young again. The egg is never really fertilized. So the egg is taken, it's like an egg extraction. So it's a haploid egg, right? So it never fertilized, it never saw sperm. And through this fusion, what you do is you introduce a diploid nucleus into it. So there was no fertilization per se. This egg goes from enucleated to having a diploid nucleus in it, and it thinks, oh, I must have been fertilized because now I see two chromosomal complements. That's the word. I was thinking components, complements, right? So it was never truly fertilized. There was never fusion of sperm and this egg. It's just the fusion of the somatic cell with the enucleated egg, and you create these hybrids, these fusomes that we'll think have been fertilized. The difference, in a way, going back to your question, the difference between these and IVF, in vitro fertilization, is in vitro fertilization, we take a real egg and sperm in a test tube, we put them together and we let normal fertilization go through, and then we take those fertilized zygotes and introduce them back into the female animal. That's IVF, but this is cloning because you're taking the nucleus out of the egg and introducing a fully formed nucleus. At some point, this cow was made transgenic through genetic engineering. So there was at some point a petri dish with cells in it, and we introduced a transgene in it, and we could follow that transgene along the way with PCR and fancy techniques, and we have really good control over how this transgene is now going to go into all of these cows, all of which are going to be transgenic. What we don't have a lot of control over is what's happening to other genes in that genome throughout this process, right? Will these fusion, for instance, create some sort of stressful environment within the cell that will induce some mutations? So normally what happens is before you put this into a cow, you can check for certain markers, for like you can do some experiments, because what happens is you don't normally inject this thing. You let this in a test tube grow into an early embryo, and if it made it past the first few cell divisions and it forms some of the early embryonic structures, then you're confident that things should progress normally the rest of the way, and then you inject them. The mammary cells are the ones that Ian Wilmot used as somatic cells for making Dolly. And the story goes, and I kid you not, that because he used mammary cells, he called Dolly after Dolly Parton. All right, so I want to get through the rest of today's lecture, which deals with how we make transgenic plants. All right, so some of the concepts are going to be very similar, but the technology is a little different for a very intuitive, simple reason. It's not that easy to put things into plant cells because plant cells have a cell wall. It's a little harder to introduce things into plant cells experimentally. So there's been two solutions. One we'll study in more or less detail in the next few slides, and that's using a bacterium that normally injects its DNA into plant cells. So it has the machinery for injecting things through that cell wall. It's called well, I'll show you in the next few slides. It's using these two, the TA bacterium, the Agrobacterium tumefaciens, that's it, AT, not TA, AT. The other solution is a little less intuitive until you hear about it. And it's called bioballistics. And some of the reading material in Canvas goes into these in much more detail. But it is exactly what it sounds like. You use a biogun to coat tiny microscopic molecular size pellets with your DNA of interest, and you shoot them into these plant cells with enough force that they will penetrate through the cell wall and hopefully stay somewhere hopefully in the nucleus, right? So you shoot them and you hope that some of those DNAs will make it into the nucleus. Once inside of the nucleus, the usual trick is the same. You hope for some sort of homologous recombination that will allow that plant cell to incorporate a piece of foreign DNA into its own genome as if it were its own. One of those sort of emergency reactions in which the plant cell nucleus sees this floating piece of DNA and it says, oh my God, we need to put this back. Where should we put it? And then it uses some homologous sequence to its own genome to mediate that homologous recombination, then introduces it in a location that you, in a way, planned in advance. Okay? But let's talk about Agrobacterium tumefaciens. Agrobacterium tumefaciens is a bacterium that is, again, a natural bacterium, seeks in nature and lives in soil. And what it does is it infects plants through the roots. And what it does is the following. It has its own bacterial chromosome, shown here. So it's a bacterial chromosome, large, circular, closed, like a regular bacterial chromosome. And then it has a plasmid. That plasmid we call the TI plasmid. And that TI plasmid has a bunch of very unique enzymes that allow this bacterium to create a molecular needle of sorts that it uses to inject the TI plasmid into the target plant cell. Once inside of the plant cell, these TL and TR sequences that you see there, I'm going to do it a little more slowly, TL and TR sequences that you see there, serve as sequences for transposon-like recombination. So there will be some recombinases produced by the TI plasmid that will use the TL and TR sequences to introduce anything between them into the host plant cell, okay? Or the infected plant cell. So far, so good? So it's like a plant transposon that it injects itself in the form of a plasmid and mediates the recombination of anything that is between the TL and TR sequences. Naturally, what's between the TL and TR sequences are tumor-inducing genes. So these genes will confuse those plant cells and make them divide and divide and divide and divide and keep dividing. And in doing so, they will create an environment that is advantageous for the bacterial cell. Because now there's going to be all of these nutrients that come to this region of the soil. The plant is creating this tumor underneath the soil, right? And bringing in all of these nutrients and sugars and biomolecules, and the bacteria can use them. So it's a sort of a parasitic relationship by which the bacterium is inducing the formation of a tumor in the plant and benefiting from it. In the lab, what did we do in the lab, back in the lab? Someone took those TR and TL sequences and used them to flank a regular multiple cloning site in a plasmid, in a vector. So now anything that you put in that vector will be flanked by TL and TR. So any transgene of interest that you're introducing in this vector will now be flanked by TL and TR. Now, you want to create a transgenic plant. You don't want to create a tumor in the plant. So what the people do in the lab? Separate the injection machinery from the recombination machinery. We took out the tumor-inducing genes already because we replaced them with our transgenes of interest. But what we don't want is the whole TI plasmid going into the plant cell. We only want our transgene of interest to go into the host genome. So these modified Agrobacterium tumefaciens cells that people use in labs will have a helper TI plasmid that produces the recombinase and produces the machinery for making that molecular needle. And then you have your plasmid vector that the only thing it has is the sequence of interest with the TL and TR flanking it. So when these modified agrobacterium cells inject these plasmid into the plant cell, there is only in the recombination and incorporation of your sequence of interest into the plant genome. And there is no longer a tumor because you've taken those tumor-inducing genes out of the natural plasmid. So I'm going to go through these kind of quickly. The way this works is you take a piece of a plant that you would like to make a transgenic out of. You incubate that piece of plant tissue with your modified Agrobacterium tumefaciens, meaning the one with the helper plasmid, and then another vector with your sequence of interest. You let this modified Agrobacterium inject the vector into plant cells. Most of them are not going to be injected because, like with any infection, there is a certain degree of randomness here. So most of them are not going to be injected. You can see them here. So that one is not injected, this one is not injected, that one is not injected, that one's not injected. And those that are injected with your vector, with the sequence of interest, many of them will not really incorporate it into their genome because, like, then again, these recombination events are not foolproof. They're not 100% efficient. So there's yet another layer of randomness here, or probability. But there may be one or two cells that did incorporate your transgene of interest into their genome. And what else goes with your transgene of interest? A selection marker. How do you kill plant cells? Herbicides, that's right. So what you do is, along with your transgene of interest, you introduce some sort of herbicide resistance gene. Think Roundup and Monsanto crops, right? That we use Roundup to kill weeds, but then we make certain plants resist Roundup, so farmers can grow plants and spray Roundup like crazy because they're going to be killing everything except their crops. So that's sort of the idea. So those cells that incorporated your transgene of interest and the resistance to the herbicide will keep proliferating and will form something we call a callus. And that is shown here, like one of these things in light green. It's just a pile of cells that keep proliferating in vitro, right? And that have your transgene of interest incorporated into them. And when these calli, that's the plural for callus, when these calli keep proliferating, or calluses keep proliferating and proliferating and proliferating, eventually they will start forming plant tissues. Here in this cartoon, it's callicili because it forms those leaves, right? But most likely there's going to be first some root shoots and then some sort of meristem for the stem and so forth, right? But this is cartoonish. The idea is that from these calluses, you can get a plant that is a clone of that one cell that incorporated your transgene of interest into its genome. And what will that transgene of interest be? Well, anything you wanted to put in a plant. It could be a gene for resistance to droughts. It could be a gene that makes the plant flower earlier in the season. It could be a gene that you know is associated with making larger tomatoes. I don't know, whatever you want in that plant.